Thank you very much for the kind introduction. Thank you also for inviting me and our company to this event. My name is Schwarz and I'm the Managing Director of Numaferm. We at Numaferm, we focus on peptides. These are more or less, how to say, small protein fragments. So they are composed of amino acids as proteins and enzymes as well. However, they are much smaller, so they have distinct characteristics and functionalities compared to proteins. And having said that, peptides are biomolecules, and we at Numaferm, we are specialized in producing these biomolecules by a biological approach. And that's why we call it biomade peptides, as shown in our slogan. We try to cover a diverse range of services across the entire value chain of peptide-based drugs. So we have discovery services in place at Numaferm. After having defined lead candidates, we go to the process development phase, where we try to develop processes that are scalable and very cost efficient. And after having done that, we try to we handle the production of these peptides at various scales and quality gates, like, for example, GMP grade peptides for the pharmaceutical sector. We also handle ISO certified productions, food grade productions, and up to the ton scale if this is needed by our clients. By doing so, we really focus on, how to say, driving change in the peptide world, and this is and we do that by unlocking the potential of peptides, because nowadays there are distinct limitations during or with peptides and during the process from becoming from a discovered lead candidate to the production, and we try really to solve these issues and unlock the power of peptides by doing so. Where did we come from? So Numaferm is a spin-out, as heard before, from the University of Düsseldorf. We were officially incorporated in 2017. However, the core team came together in 2015 already. As you see here, before this time, already close to 10 years of work happened to come to this stage. Actually, the start was kind of my PhD thesis at the University of Düsseldorf, where I found surprising good results during my studies. And having found these data, the University of Düsseldorf, together with my institute head and other people, we filed patent applications. And what you can see now here, we continue to doing so, and we are really nowadays an innovation-driven company with more than 30 patents applied or granted. In this time at the university, up to 2017, we did first reference processes with clients from the industry, and very importantly, we convinced investors to trust in us and to invest in us. And having these investors on board, we moved out of the university into our own lab space. We started with a few square meters. Actually, nowadays we have a facility of roughly 500 square meters in the Life Science Center in Düsseldorf. Since then, from my point of view, we did quite good progress. So we have happy clients from my point of view. And just recently, like a year ago, we had like an anniversary where we produced 200 successful peptides. So we really did a lot of different kind of peptides already. We closed the first license agreement with one of our clients, and we were able to show that our technologies are really industry grade by upscaling them in the multi-thousand liter scale, in the kilogram scale of the products. And we also got an ISO 9001 to 15 certificate to demonstrate that we have become a mature company. But what are we really doing in comparison to what is done nowadays? And to understand that, I have prepared this slide, how peptides are nowadays produced. So what's the state of the art? And actually, Mr. Merrifield got a Nobel Prize in 1984 for these developments, how to produce peptides. It's called solid-state synthesis. And since then, this method is really the dominant approach to produce peptides. Even nowadays, more than 85% of all peptides globally are produced with this or adapted approaches. And this is a really reliable approach that helped humans a lot because peptides got accessible. However, there are some limitations, challenges with chemical synthesis, and this is the cost of the production. If you want to produce 1 kilogram of a pharma-grade peptide, this easily costs €1 million. And the explanation for that is that you need just so many raw materials. If you want to produce this kilogram, you need 25 tons of raw materials. And of these raw materials, part of these raw materials are organic solvents and really harmful, toxic chemicals. So it's a really, really good approach, but maybe it's a time to have an alternative or at least an alternative to this approach. And this is what we have developed. Our approach is a biotechnology-based approach. Biotechnology is a very mature technology per se. However, today biotechnologies are made to produce proteins and enzymes. And chemical synthesis is used to produce peptides, like these small protein fragments. We have now developed a technology that can produce peptides recombinantly / by biotechnologies for the first time reliable. And this independent of the length of peptides, the characteristics. And we do that in a way that is really sustainable compared to chemical synthesis. And we get highest qualities of peptides, increasing safetyness of the products. And so that's very good also for the customers. If we need to figure out a few core USPs, so unique selling propositions of our technology, I would choose the one that are shown. Very important are the cost of goods that we reduce. So we can show that we can reduce cost of goods by more than 90%. As I mentioned, it's not a chemical synthesis-based approach, it's a water-based approach. So we don't use organic solvents, which is in the green economy, I think, a very, very important factor. Also, we don't use harmful chemicals. It's a bio-based approach. So there's nothing that harms nature. And it's a scalable approach, and that's very important. Besides being very expensive as raw material, peptides can also not produce in huge amounts that are needed for many applications. For example, if you consider peptides being applied in the agriculture sector, you easily need tons or hundreds of tons of such peptides. And chemical synthesis just does not allow that, for example, because you need so many raw materials. It's just not possible. And overall, we can show that we can reduce the carbon footprint by more than 90%. Our services are summarized here. So I had a brief overview already. This is here a little bit more detailed. So the discovery service is to identify your new leads. So we want to give our clients the opportunity to get new peptides in their hands that they can then use to develop new drugs or other raw materials. When a peptide is defined, we have a service called cell line development. So we do the programming of the cell. I will have a few words on these timelines related to this service offerings on the next slide. After having the cell line in our hand, we need to develop a process. So the cells are producing the peptide, but that's not all, of course. We need to squeeze out somehow the peptide and get a quality that is accepted by the relevant market. And you can imagine the quality needs to be very high in the pharmaceutical market, for example. That's done in the process development phase, and then after having done that, we have a very scalable process. We have delivered our clients test material so that they can validate the material, and then the production at scale is managed by us. This is something that is relatively detailed, I know that, but it's important for our clients, so I didn't want to not having a statement about that. So when you talk normally about biotechnologies producing proteins, enzymes, or peptides, it takes really, really long time. So you can expect development times from 18 to 24 or maybe even 30 months. And we really cut down these development times and simultaneously the development cost dramatically. So the cell line development phase just takes a week. You can imagine we cannot spend so much money to work 1 month. So it's just really cheaper and quicker than others can do that. Then the optimization, upscaling part and development of analytics need to release a peptide. That takes roughly 2 months. So overall, we have the process set after 3 months. And if you have, as I mentioned, with alternative biotechnologies, this just takes longer. But with chemical synthesis, it's in the same time range. So what we found was it is really important to offer these time schedules mentioned here, these quick time schedules, because alternatively, it's very tough to convince clients not to go with chemical synthesis because speed is money, you know, and if there is an approach, even if it's harmful and expensive, you need to choose what do you go for speed or like these other aspects. And we can really give them the material in a comparable time frame than with chemical synthesis. And this is really a clue what we found, why our business model works. And after having done the process development and the test sample supply, then, as I mentioned, we go to the GMP or commercial scale production. Overall, we talk about a project that is fairly below 1 year in overall time. And this is quick. Maybe it's not so understandable, but that's quick in our business world. These are technical data for those who are interested in the industry grade of the technology. So we have shown that our fermentation processes are very reproducible. We can transfer these technologies to other third-party manufacturing sites, and all data are really reproducible, so that's important. We have clients and projects running in quite different market sectors shown here. Nowadays, peptides are more or less only present in the human health sector due to the cost of goods of the raw material. We now talk and work together with clients from totally different areas mentioned here. I will just show that. So this is data for people that are interested in working with that, what can be expected, and to say that in a very short way, you can expect that we can produce peptides independently of their length and physicochemical characteristics. You can expect a very, very standardized upstream or fermentation part, just takes 1 day. You can expect a very easy and cost-efficient scalable downstreaming processing to get peptide in a high quality. Overall production times are only a week, and this is really a difference to chemical synthesis. If you think about producing peptides chemically, and they are like a complex molecule, this can take months for 1 batch. So this takes with us only 1 or 2 weeks. And you can expect gram per liter. Liter means fermentation liter, titers, and highest purities up to the API quality grade. This is a summary of clients, customers, partners that we work with together, statements of people that worked with us so that they are convinced by our service offering. Evonik, for example, is even invested in Numaferm. So we have done quite a lot of things and learned so much in this project. And yeah, we are really happy to have these happy people. This is a structure of Numaferm. So we are a venture capital-backed company. Evonik is invested, the high-tech Gründerfonds, the European Investment Fund. We have projects running funded from the state, Germany. And shown below are 2 business angels of us, Mr. Schumacher and Mr. Riesner, both co-founders of QIAGEN. And also we have all the experts needed in our advisory board shown here. With that, I would like to thank you for your attention. I hope I could make the point somehow how we believe that we drive the change in the peptide world. of course, if not, I'm happy now for questions in this context or in whatever kind of context you like. Thank you very much.
Yeah, many thanks. Congratulations to your development. I think you are faster, cheaper, higher volume and smaller footprint. So it's no wonder that your clients are very interested and investors too in your development. Before I have a few questions. Before I start the question, I want to mention that you can place your question with the Mentimeter web link you can find on YouTube and the specific code. And I also want to mention that the whole event will be recorded and you have access a few days later. You have the full access to the event. Yeah, the questions from the audience. Can you give specific product examples for the different application fields where peptides are used? You talked about pharmaceuticals, you mentioned agriculture, cosmetics. I think this is a very interesting point where you're looking for which application you're looking for.
Yeah, one application for sure is the pharma sector, and there we can reduce cost of goods, especially or in particularly for the generic and biosimilars market, because there the cost issue is more significant than in new development phases. But in both areas we are active. Another very interesting characteristic of peptides is the antimicrobial behavior. So we have projects running in quite different market segments mentioned before, where the antimicrobial activity is considered to substitute for antibiotic usage, for example. This is really like something we feel from the market that's traction that exists.
You mentioned also agriculture. Is this also a field? Because this would be a high volume application.
It's a high volume, yeah, and we also have a project running in agriculture where we really want to treat fish with antibiotic-less possibilities by peptides.
Yeah, to make aqua farming really sustainable.
Yeah,
it's not the case today. Many cases. Yeah. Okay, I have a question to your raw material. What is the feedstock of your process?
In our fermentations, for example, we use so far like pure glucose or glycerol. So we don't use like shredded maize or something like that. We really use these pure pharma-grade carbohydrates.
Yeah, okay, yeah. There are many sources for them, of course. Yeah, there's a question, what are the next consequent steps for your company?
Yeah, so we believe that in the last years we really developed a very mature technology that was demonstrated to work. The next step is to scale Numaferm up, so really to grow, and it's very important to be known in the market to get such service contracts. So we really do business development, marketing, sales more and more at the moment to grow the business.
Is the peptide development a worldwide topic for biotech, or do you do a special topic here in Germany, or how you see the international situation about peptide research?
Being a German company, we see in our client structure a preference to German and close by European countries, just to the fact that we are located here. But in general, it's worldwide business in Asia, the US and European. The pharma sector for peptides, for example, is the biggest one in the US. We have projects running in South America, in the agri sector, for example, so it's really worldwide.
And if you look to the, if you want to expand and grow as your company, do you see enough support in Europe and Germany for this step, or are you mainly depending on partners in industry, or is there also some policy support for growing?
We are very happy where we are in Düsseldorf, in the state North Rhine-Westphalia, in Germany and in Europe. So we cannot complain about that. We have a strong shareholder board, I believe, and of course they are supporting us so much. We have the know-how. Of course, Evonik also have infrastructure where we can have hands-on during projects. So we have nothing to complain at the moment. Yeah.
And the access to risk capital also went better meanwhile, what I heard?
We didn't have these issues, okay. But we as company did not raise money in the 2-digit million area so far. So we were in the 1-digit million area, and this worked quite well. So I don't know what would happen if we go for the bigger money, but at the moment it worked very good.
You mentioned also the business angels. Was this important for your development?
I believe it was very important because our business angels, they really trusted in us very early, and we could go with their commitment to VCs, venture capitalists.
Yeah.
And this makes us more mature, and I think was one of the keys why we succeeded in, for example, convincing high-tech Numaferm, but also Evonik.
Yeah, also to avoid some mistakes that often happen with young companies. Yeah, I have the last two questions here from the audience. One is, how is the carbon footprint reduction exactly achieved?
So when we produce our peptides, we can show that we don't use 25 tons per kilogram as by chemical synthesis. We are in the range of 1 to 1.5 tons raw materials, mainly water and nutritions like vitamins or, as speaking before, carbohydrates. So this already is more than 90% reduction in the carbon footprint. But on top of that, we don't use organic solvents, or only minorly if the peptide needs that for its personality, we call that. We don't use toxic chemicals that needs to be discarded in a specific way. That's the answer on that.
Okay. And in addition, we have to go in the LCA in detail to understand the different steps. But okay, if you can really save already as a feedstock so much CO2, then of course you are on the safe side. Yeah, we have a question on biomaterials. Can your platform be used to scale biomaterials like collagen or more exotic peptides like the squid ring teeth?
Yeah. So, for example, what was keratin? What was no? It was
collagen.
Collagen.
Yeah, collagen. Yeah.
So one thing that needs to be understood about our technology is that we use E. coli to produce material. And E. coli has like a spectrum of amino acids that can be incorporated, namely the canonical or the natural amino acids. If we think about collagen, for example, there's a hydroxylation reaction of some amino acids. So this is a non-natural amino acids. What we can do at Numaferm is we can add non-natural amino acids during the downstream process by chemistry, by enzymatic reactions, like by click chemistry. That's possible. But our raw materials are consisting only of natural amino acids. That's why we are called as bio-made peptides. So we do biopeptides and not of non-natural amino acids, despite the fact, of course, that collagen is a natural product, but transformed in the post-translational way. So we are focused on natural amino acids, but do all the tricks that is state-of-the-art in the downstream to come to such moieties.
Okay. So I think that's pretty clear, and many thanks for these insights, and congratulations and good luck for the future.